Good afternoon, everybody. Thank you for joining, and again, thank you to the organization and ARM at the MESA meeting to allow me to present. I'm Miguel Fort, currently the CEO of Kiji Therapeutics, but also heavily involved in ARM. What I would like to present to you today is a gene therapy through a cell delivery, taking advantage of a lot of experience and bringing that to today's standards, delivering increased benefit for unmet medical needs. You will see there that Kiji Therapeutics has three elements: iPSC, gene therapy, and MSCs. Why do we need a new paradigm, a change for an approach with MSCs? We're ready for that new paradigm because MSCs have unfulfilled efficacy and manufacturing limitations. I don't need to recall the multiple experiences on that, but those experiences gave us the background that can enable us to then bring the value that I want to talk to you today. And the solution is to really engineer those cells, genetically engineer for targeting and for potency, and deliver through a proprietary gene sequence that we have a opportunity for autoimmune diseases, starting with GVHD and IBD, and using an iPSC technology to also improve. So we're actually addressing two aspects: the manufacturing through iPSC, improving that aspect, and the potency through genetic editing. It is game-changing. It is off-the-shelf opportunity based on the elements that I mentioned to you. We're well advanced. We are just over a year from being in the clinic with a network of support and physicians in Spain. And we are supported by a group of founders, excellent management, and all the capabilities that would enable us to deliver on the opportunity. So what do we represent? The mission, in other words, what we want to deliver is develop off-the-shelf, iPSC-based therapies for life-threatening diseases. We have an initial product that is lenti-transduced with IL-10 for potency, CXCR4 for targeting. We're on track to enter the clinic, first patient to be dosed, before the end of 2024 in refractory GVHD. And this study will be the first study that will deliver evidence on the opportunity of engineering these cells for optimal potency, and so would be the validation, the proof of concept of the approach. A much needed opportunity for MSCs, I would say. But we will go beyond and have a tool sourcing the MSCs from iPSCs and engineering them for efficacy, as I mentioned. So really providing a genetically modified iMSC for transformative therapeutic efficacy. We've been created in France this year, so just over 6 months ago, and we work between Paris and Madrid, where our founders are based. We have an initial seed from a group in Paris, and we're looking for an extension of that seat. The management team brings a lot of experience, and you may know some of the faces there. And we're supported by an expert international advisory board picked to bring experience on iPSC, MSC, engineering, and the clinical indications in hematology and IBD. The principle is very simple. We engineer the cells by transducing the CXCR4 IL-10 that you see pictorially there on the surface and around the cell. And in that way, we will deliver a transformative iMSC approach. This approach in the preclinical data that we have and is extensively published has been very validated. In other words, there's clearly no doubt that engineered cells have an increased opportunity of efficacy in multiple animal models and repeated animal models. Obviously, the proof of the concept will be to take it into the clinic, which we aim to do. And that is based on potentiating a key element of the mechanism of the MSCs. So in a way they do this, but we intensify it by the transduction of the gene. CXCR4, as I said, for targeting, providing the MSCs to go to the focus of the inflammation and there deliver the immunocontrolling IL-10, which is one of the key drivers of the mechanism of action of MSCs. We have three groups of products. Kiji 01, which is really the first one, the one that will go to the clinic, adipose-derived MSCs, the same ones that have been used for the same source for alofisel, and which is nearly ready. We're finalizing the GMP elements, namely the viral vector, and as I said, we should be in the clinic next year. Behind that, we will use the same gene, but in iPSC, providing us an opportunity to go to larger indications, and we aim IBD in that context as well as others. And beyond that, we will have additional genes, giving that modularity of iPSC sourcing, MSC, engineering, and therapeutic target. The key data is really transforming an inflammatory condition to a non-inflammatory condition, and we have evidence of all those elements. And there, you can see at the bottom the paper that actually was recently published and brings all of this data. The two graphs that you see there show the incremental effect of the two modifications that we do. On the left-hand side, we see that the gene-transduced MSCs with CXCR4, whether they have IL-10 or not, migrate more than the others. On the right-hand side, we see that the ones that have IL-10, whether they have CXCR4 or not, have a stronger immunomodulatory effect. And we have data in animal that the combination of the two provides a targeted, more potent effect. The key animal model that constitutes the basis for us to go into the clinic is this humanized GVHD animal model, where we have documented that the benefit over untreated animals of gene-modified MSCs goes above the well-known existing benefit of MSCs. So you see there in black the untreated animals, in gray the ones treated with wild type with some benefit, and you see the incremental benefit above and beyond that of the cells that are transduced to CXCR4 IL-10. This clinical evidence in terms of weight evolution and clinical score is backed by mechanistic studies done on the human cells used for the immunized model, circulating in the animals, where we've documented decrease of cytokines, pro-inflammatory cytokines, and increase of anti-inflammatory cytokines, human cytokines in circulation. We've documented in circulation increase of Tregs, and we've documented increase of Bregs in the spleen. So not only having the clinical evidence of the animal model, but the mechanistic support of an immunomodulatory effect that would back the rationale to go into the clinic. I mentioned the second indication, and we do have the same evidence of incremental benefit, and you can see there a DSS model with a re-challenge, where we see the animals that have the top score, i.e. the top quartile of improvement, being more on those that are treated with gene-transduced MSCs versus the wild type, and clearly versus the non-treated ones. It's also important to note, and you might remember some of the clinical data on some MSC studies, that there is a late benefit, a long-term benefit, suggesting that there may be an impact on the innate immune system and a modulation and resetting. So an interesting opportunity to look for. And indeed, our intention is to move to the clinic with IBD, but define well the target patient population and the approach, but use IMSCs with the proprietary gene transduced. But before that, we're going to execute a study in GVHD. I don't need to labor about GVHD. There's only one thing, is that the patients that have graft-versus-host disease, 25% of them still today, despite a couple of products fully approved for that, have no treatment. So there is a clear remaining unmet medical need where we believe that, like MSCs have touched efficacy, we can bring them with an optimized benefit and really deliver the opportunity therapeutic effect to these patients. The study is well-defined. It will be conducted in Spain. It's going to be done with off-the-shelf. The product is delivered cryopreserved, thawed at bedside, and immediately infused, making it compatible with clinical practice, making it easy to use, and really not burdensome like other approaches on cell therapy. We don't need to go into the details of that, but just to tell you that we're very well advanced and ready to execute once we have the final GMP elements completed. You can see here the production diagram, and it's interesting to see that if you look at it now with an adipose-derived donor and a gene transfer, you will see that in the future we'll have the same process, but we will source iPSCs and we will have multiple genes to be used. And we do have already undisclosed data with that approach. So we see ourselves in this progression of what we're doing today, what we'll be doing shortly, and the vision of the value that we're going to deliver in the long term by starting with adipose-derived, IL-10-CX4 transduced for GVHD, while at the same time having already an R&D IMSC CXCR4 IL-10 product available, which will then, for the near future, be transformed into GMP product, enable us to go into other indications, IBD. I actually didn't mention, but we do have other animal model diseases evidence of the same incremental benefit. Some of them mentioned there, so that we will be able to progress to those indications. And finally, with that modularity, we do have already some incoming collaborations with centers that have additional genes of interest that would enable us, again, to pick and choose and target the cells for optimal manufacturing and optimal therapeutic benefit for targeted indications. This is the progress we've done and where we are. We see ourselves being in clinical stage before the end of 2024. We will then be progressing to the second target, and we have, as I said, raised the seed and aiming to expand that before being ready to deliver clinical data that would enable us to jump on that into a Series A. You can see here the operational progress that we are intending to deliver in terms of the R&D, the GMP, which is the main element there, the regulatory interactions, and the clinical, so that we will be an industrial clinical stage, one indication by the end of next year, and a couple of additional indications in subsequent 2 years. So, what is the takeaway message? Is that we're addressing an unmet medical need in autoimmune disease with a transformative gene-engineered IMSC product portfolio. We have an established rationale, proof of concept in animal, in vivo. We are about to have an industrial GMP product and R&D IMSC product. And we have all the elements and the network to be able to deliver. And we see ourselves as, with that modularity, as a leading company on gene-engineered IMSCs for optimal benefit. And donations are welcome, and questions as well. Thank you very much.